DNA is written by evolution. It's the language of life, but it's not really a language so much as it is a code base. And evolution, of course, isn't exactly known for helpfully commenting its code. But even so, thanks to advances ranging from sequencing to CRISPR to DNA synthesis, we've increasingly learned to read, to edit, and to reprogram living things. And that capability has been transforming health, agriculture, and perhaps soon, ecology. Now, if we want to engineer ecosystems, there's one main problem, which is that there's so many wild organisms out there, and they're all struggling to reproduce. They're all competing. And if we engineer an organism to do something that we want, we're diverting its resources away from reproduction towards doing the thing that we want. That means if we raise a lot of them and we release them into an environment that we don't control, natural selection is going to outcompete our desired trait. At best, it's going to linger for a few generations at about the same frequency as we introduced it before it eventually disappears. But what if we program the organism to encode the CRISPR genome editing system next to the desired change? We're programming the organism to recursively edit its own genome. In the reproductive cells of each generation, CRISPR will edit the original version and replace it with the new one. This is a gene drive system. And because this can happen every generation that inherits one copy of the gene drive, then the trait will spread because it's inherited more than 50% of the time. It will spread through the bulk of the population. This is extraordinarily powerful because it means a single laboratory can potentially edit the bulk of a species. And that has some amazing applications. Take malaria. Now, malaria is primarily caused, the bulk of cases are due to one species complex of mosquito. That's one species out of more than 3,000 mosquitoes. And a group called Target Malaria, within 5 years of when we disclosed CRISPR-based gene drive, had developed a version that could crash the population of this mosquito in cages. Crash it to levels low enough that it won't be able to transmit malaria anymore. And that could be enough to let us eradicate the disease forever. And if we can do that, then we might be able to use it to eradicate the scourge of schistosomiasis. We could use it to tame the 8th biblical plague by programming desert locusts to remain harmless desert grasshoppers rather than forming swarms and devouring crops. We could even use it to remove the screwworm that is responsible for more animal suffering than a century of factory farming. These are all amazing applications, but we need to ask ourselves, what else could such power do? Is it safe? How can we tell? Well, we can think about this several different ways. Let's start by saying, how fast is it going to spread? Will we have time to respond? Will we notice if it's spreading somewhere it's not supposed to? And can we do something about it if it does something that we don't expect? The answer, thankfully, is this appears safe. It favors defense. It takes generations to spread, parents to offspring. It's easily detectable by sequencing. You just don't see CRISPR in a sexually reproducing organism's genome. And, as we determined very early on, any gene drive system can be overwritten and its effects undone by another CRISPR-based gene drive system. So the technology can counter itself. Realizing that it in fact favored defense and was therefore safe to disclose was perhaps the greatest sense of relief that I've ever felt in my life. But there's more than that. We can actually localize its effects so you don't have to change a whole species. That is, we can limit the number of times it can be copied by building what's called a daisy chain system or several other more recent methods. And that allows us to perform safe field trials altering just a tiny fraction of a population and seeing what happens in the ecosystem. We can even build gene drive systems that promote democracy. That is, we can make natural selection favor whichever version is in the majority. And that means that if one town wants to edit a local population, they can do so, and natural selection will keep it there and prevent it from spilling over into the neighboring town that doesn't want it. That's pretty incredible. But that's not all there is to it. Because if we develop a new medicine and your doctor recommends it to you, you can say no. You can choose not to be affected by the consequences of our research. And that means it's okay if we don't tell you about what we're doing until the medicine is available. But if we're building something to change your environment, and we don't tell you what we're doing until it's fully developed, then we are denying you a voice in decisions intended to affect your life that you won't be able to opt out of. We need more than traditional open science. In normal open science, as we think of it, we share our story once the story is done, once the technology is developed, the discovery is made. That's not the most efficient way to do science. That's just how cultural evolution shaped our scientific incentives. And for Gene Drive, we need to do more. What we need is early stage community guidance. We need to go to communities, a representative community, before we develop the application. And we need to say, here are the different technical options that we think we might be able to accomplish. Which of these do you want us to pursue, if any, and give them a voice in choosing what moves forward? This is what we've attempted to pioneer, working with the communities of Nantucket and Martha's Vineyard off the coast of Massachusetts to prevent Lyme disease. And as well with the Maori iwis of Aotearoa, New Zealand, regarding using these kinds of technologies to control invasive species. And in both cases, we learned things that we didn't know, meaning the technology ended up safer than it would have otherwise. And we found that many people who really don't like the idea of engineering wild organisms, people who oppose the technology, still support the project because they support the process. Everyone agrees this is how science should be done. And if that's so, if that gives us faster and safer science, why don't we do it more often? And the answer is, we're too bloody well afraid of getting scooped. Science would be better off if we change those incentives to favor early disclosure of our plans. How can we do that? Well, maybe Gene Drive can be a field trial. If the WHO were to sponsor a registry of all Gene Drive experiments that required community sponsorship and early laying out of safeguards, then journals and funders could require it, and we'd change those incentives, and Gene Drive could be a field trial of truly open, early-stage, community-guided science. Now, that's pretty amazing. But we also need to keep in mind that exponential biology has a dark side. Not everything that can spread on its own is good for us. And of course, we've just lived through this. Pandemics do not favor defense. Omicron spread to infect half of Europe within 100 days of being sequenced. Even the ancestral strain could spread without symptoms. And of course, it was far too long before we could do anything to save lives. If another pandemic began today, we wouldn't be able to stop it from killing millions of people. And that needs to change because we will likely learn to cause new ones. This is Richard Feynman, and he said, "What I cannot create, I do not understand." Our very desire to understand how pathogens work, to more effectively program living things, will eventually teach us how to build pandemic-class agents, to edit existing things, to make them more transmissible, to make them more virulent. It will be inadvertent. It will be a byproduct of our attempts to build defenses. but we will learn how all the same. And today, thousands of us, many people in this room, I'm sure, have the technical skills to assemble infectious viruses from synthetic DNA. The answer to all of this is to ensure that we have those defenses prepared before we learn to cause new pandemics. What does that mean? Well, we can limit access by requiring companies to screen all synthetic DNA for hazards, particularly potential new pandemic agents. A new Swiss nonprofit foundation, the Secure DNA Foundation, which is a collaboration between American, Chinese, and European biologists and cryptographers, will this year begin offering free DNA synthesis screening, cryptographically secure, to all providers throughout the world. We can ensure that we see something coming, no matter what it is, even a fast respiratory equivalent of HIV. If we sequence wastewater and look for signatures of exponential growth, which particular sequences appear over and over again in increasing frequency in those sequencing reads, any threat must grow exponentially if it is biological and is to threaten the world. We will see everything coming. And of course, if we really want to block a pandemic, you want to ensure that all essential workers have the equivalent of powered air purifying respirators that will keep them safe during the next pandemic while they keep the rest of us alive. But perhaps most promising of all, since none of us really wants to wear bubblehead helmets, is we have lights. What's called far UVC, 222 nanometer lights, it's germicidal, and it doesn't seem to affect us in any harmful way whatsoever. And the reason is it's absorbed by proteins, so it doesn't penetrate our eyes and skin, but it still destroys microbes in the air. And currently approved levels are more than 10 times as effective as an aircraft ventilation system on full blast. Install this in public spaces everywhere, and we might not only stop future pandemics in their tracks, but we could potentially end respiratory and surface-transmitted infectious disease. All of it. In the end, technology is power. And, of course, with great power comes great responsibility. If we can be humble enough to acknowledge that there is much that we do not know, if we can invite concerns and criticism from others before we make powerful new advances, then we might just be able to build a wise and flourishing future for everyone. Thank you.
I'm actually going to go to Francoise Bayless, if I may. Francoise is the ethicist who is on the Science Council, but there was a very good suggestion you had that actually funders shouldn't be funding unless there is already evidence of community consent, for instance. Francoise, your thoughts on this, and can we get her a microphone? She's there in the front row.
Hi, Kevin, how are you? So I guess what I would ask you, given that I think we're both committed to public engagement and consultation, if we take your last suggestion with respect to the lights and what it might do to make our lives better, how would you imagine doing that global consultation at this point in time?
Ooh, that is a fantastic question that we're grappling with now. So the question is, how high can you go in the intensity of these lights? And how do you ensure that everyone believes that they're safe? And as best you can tell, the way forward is to bring together everyone from all the different fields that touch on it, ask them to poke holes in our current understanding, identify any gaps, and lay out a plan of experiments to address them, then make that public and invite everyone to take potshots at those plans and say, nope, you're missing this, you're missing that, this experiment isn't enough, this needs to be replicated this many times. And then we need funders to actually fund all of that. Because this is a case where we probably only get one shot. I mean, the thing is, we know that from GMOs and certainly gene therapy and arguably vaccines, the backlash can set back a technology by a long ways. And that is a real risk these days in the age of social media. So we have to be supremely careful. We can't afford any mistakes that would result in inadvertent harm. And we have to recognize that these kinds of technologies are not going to be developed because people want to prepare for the next pandemic. I might care about that, you might want to care about that, but the world wants to forget. So we have to develop it and ensure that it's safe enough for people who only want to use it to block run-of-the-mill infectious disease, who don't want to get the flu, who don't even want to get the common cold. That's the way I think that we have to move forwards. But again, I'm not the expert here. You are, and what's more, all of you are. So this is a case where we really need to invite concerns and criticism, even about the process.
Can I take a question here?
Hello, I'm Nova from Bangladesh. I actually had the same question for the previous speaker, because when we come from a developing country and we talk about the voices, which I really appreciate, sometimes just having the voice is not enough, because if we reach to a community, possibly these are the communities getting the vaccines through universities, actually through companies funded by different vaccine companies. So the knowledge inequality will play a role. So I was wondering what you all think about that, because when we want to open science, we have to actually open borders, open the thought process as well. Thank you very much.
That's a fantastic question, and I think that's another area that ties back to funding. We can only do this kind of engagement if the funders actually provide us money to do that. Right now, there's just no positive incentives for us to do this other than that we know it's the right thing, as you described. But it's also an area where, frankly, my group is on the cowardly side. Target Malaria is out there working with local African communities, ensuring that the concepts can be as effectively as possible translated into local languages, and inviting people to weigh in on how they would like the project to proceed. Whereas we started in Martha's Vineyard in Nantucket, which doesn't have any of the power dynamics issues because these are some of the wealthiest and most politically powerful communities in the world. There's just no way we could push them around or make them do anything. Everybody speaks English. They have tradition of New England town hall democracy, and of course, just about everybody there knows somebody with the technical background to understand the details of how we're proposing to edit these mice to block Lyme disease.
That must be really scary.
Right? Like, well, no, it actually makes it very easy because like one person in Nantucket said, oh, well, I really don't like the idea of engineering the mice. That's just awful. I really don't like GMOs, and I really don't like the idea of vaccines, and editing living things is just something we shouldn't do. But I'm not really an expert, and my friend has a PhD in ecology and really knows the island really well, so if she thinks it's a good idea, I'll vote for it. If only.
If only. We're going to finish with a question here, which has come from someone else in Bangladesh. Hassan Mahmoud Reza, who's Dean of the School of Health and Life Sciences in Bangladesh. Can gene editing technology be proved safe and effective through RCTs? Small molecule safety and efficacy against diseases established through RCT.
Okay. So this is a regulatory issue that regulators have experience with RCTs. The C is clinical, right? But for an ecological technology, you have to use a field trial. So while clinical trials are fine for gene therapies and things that are geared towards the clinic, most of the regulatory agencies don't really have experience in ecology. And yet what we've found is that our mice, for example, are going to be regulated by the Food and Drug Administration, because it turns out it's a veterinary drug when you change the genome of an organism, even if it's heritable, because you're changing the properties of the organism. And so the FDA will be overseeing field trials and looking at ecological effects. But the Center for Veterinary Medicine there has been very good and open-minded about it. They just made it clear that, sorry, for political reasons, it's got to be us, but we'll loop in EPA and the other experts to try to make it as smooth as possible. But it's a real problem, and I think overall the same principles apply. You do need, it's not quite a randomized controlled trial, but you do need a small contained environment, uninhabited islands, for example, where in one region you employ the intervention, in another you do nothing, and in another you introduce new organisms that are wild type to just see whether any effects are due to introducing new organisms.
Kevin, you've got a lot to do, but one final thing. In one phrase, how would you describe what an exponential biologist is?
It's someone who thinks about the potential power of things that can spread on their own to shape the course of life on this planet.
Now you know, and there'll be a test later. Thank you so much, Kevin.